The overall goal of this procedure is to directly visualize and quantify the activity of DNA-binding proteins in live bacterial cells. This is accomplished by visualizing cells that express a photoactivated fluorescent protein fused to a DNA-binding protein of interest. The second step of the procedure is to acquire a movie of individual proteins getting photoactivated while being imaged. Then, the data is analyzed to determine protein localizations and track proteins inside single cells. DNA binding events are identified by change in the diffusion coefficient of single proteins when they contact chromosomes. Ultimately, the results can provide a quantitative measure of protein-DNA interactions at the single-cell level by counting the number of bound and diffusing proteins per cell.
This method can help answering key questions in the DNA repair field, such as the in vivo repair rates and the spatial distribution of repair sites. Now we'll demonstrate the method by measuring the repair activity of DNA polymerase I in live E. coli cells.
First, make cover slips with no background fluorescent particles. In a furnace at 500 degrees Celsius for an hour, burn a stock supply of cover slips of thickness number 1.5. Store them at room temperature in aluminum foil. They are good for weeks to come. Next, concentrate a milliliter of early